episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show. We're bringing you another fascinating guest today, helping to create a better tomorrow for all of us on many different fronts. We have the honor today of being joined by Dr. Eric Verdin, President and Chief Executive Officer of the Buck Institute for Research on Aging, as well as a Professor of Medicine at University of California, San Francisco. Dr. Verdin's lab has been involved in studying the relationship between aging and the immune system, for studying the various associated defects in both the adaptive immune system as well as chronic overactivation of the innate immune system. He also has programs there on how metabolism, diet, small molecules ultimately can help regulate the activity of various targets like sirtuins, histone, deacetylases, and thereby accelerating the aging process and associated diseases, including Alzheimer's. Dr. Verdin has published more than 200 scientific papers, holds more than 15 patents, is a highly cited scientist, and has been recognized for his research by both a Glenn Award for Research in Biologic Mechanisms of Aging and a senior scholarship from the Ellison Medical Foundation. He's been elected member of several scientific organizations, including the American Association for the Advancement of Science, American Society for Clinical Investigation, and the American Association of American Physicians. Also serves on the Advisory Council of National Institute on Drug Abuse at the NIH. Dr. Verdin has extensive experience working with and founding biotech companies, and he has advised and consulted to the numerous pharma companies, including Novartis, GSK, J&J, the list goes on and on. Dr. Verdin received his medical degree from University of Liège, completed his clinical and research training at Harvard Medical School, and has held faculty positions University of Brussels, at the NIH, and at the Picot Institute for Medical Research. We have a lot to talk about today, but Dr. Eric Verdin, thank you so much for coming on the show.
Good to be here, Ira. Thank you for having me.
Absolutely, absolutely. I'd love to start off with a quote. Science is showing that while chronological aging is inevitable, biological aging is malleable. There's a part of it that you can fight, and we're getting closer and closer to winning that fight. Sounds familiar? Dr. Eric Verdin. Eric, I'd love to start off by handing you the floor for a couple minutes just to talk a little bit more about how, throughout your career, focus on amazing topics like microbial pathogenesis, HIV, lymphocyte biology, immunology. When did aging in that history become an interest of yours?
So I think my career is one of happenstance and lucky events. And the way we got into aging was really by chance and somewhat by circumstances. So my lab was working actually on the molecular biology of HIV, trying to understand the transcriptional regulation of HIV. Yep. And we were one of the early lab in the epigenetic space. And our contribution at that point was to realize that HIV was regulated by a series of proteins called histone deacetylases, HDACs. And so my lab cloned many of these proteins back in the early 90s. And we were busy studying epigenetic regulation, basically in the context of HIV transcription. And I had a new student who came in the lab, and at that time, Lenny Garante had published an amazing paper showing that a protein called SIRT2 in yeast, which was suspected to be an epigenetic regulator, turned out to be also a histone deacetylase. And so with the new student in the lab, we went and looked for SIRT2 homologs or orthologs in the human genome, and we found sirtuins. And so we were the first one to clone the human sirtuin genes, many of them. And so that ended up being sort of both a new project in the lab, but also one that really took us into the direction of understanding the role of epigenetic regulation in the context of aging. And so a big part of my lab has been busy doing this for the last 20 years, really characterizing many of these sirtuins and understanding what their functions are, and really trying to put them into the whole picture of our global understanding of aging.
It's interesting in talking about that global understanding. I was watching a presentation of yours from, I think it was last year's Aging Research and Drug Discovery Conference, and you put up the slide with the hallmarks of aging, and you make this really important point that we believe that ultimately, if we solve all of these 9 things, that we're going to have a beautiful future, but we never really study synergies, the hierarchies, what's additive, what's not. I'd love to start off because it was, such a major announcement just a couple of weeks ago. The Buck Institute with another group called the Astera Institute announced this $70 million collaboration in the field of aging, but you go into the small print, and you mentioned there's little to no research in combination therapies, although most people believe that, hey, we got to start combining these things, putting them together to really leverage the synergies to solve this health span issue. It's interesting. Early in my career, I had some combination sort of therapeutic development experience. And I know sort of back in the 80s and 90s, let's say, if I went to any of your partner companies that you worked with in the past, whether it's Novartis, GSK, wherever, if you told them you're developing a synergistic combination, they kind of stare at you. But we now have regulations, things on the books at FDA, EMEA, and so forth, where there are now plans that, hey, if you want to bring us a combination, this is how you do it. Talk a little bit about this deal, if you would, and then if you could talk a little bit about this whole concept of combinations, of synergies, and why it's really so important to understand, if we take an mTOR inhibitor and a senolytic and a stem cell, why we have to understand how they're all going to be working together.
Yeah, so it's a really good question. So let me back up maybe and just restart with the hallmarks of aging, because in some way, the hallmarks of aging was a really key paper in our field, and it's been criticized that this is an incomplete understanding. And I think looking back and criticizing is easy. I think what I think we have to applaud is the people who wrote the paper and actually provided a framework for the field to organize itself. And I think what was important in the paper was really the identification of a number of key molecular lesions or pathways that are affected during aging.
Surely,
and essentially, sort of guiding us to where are the areas in which we should be focused. There was one big omission in the hallmarks of aging, which was chronic inflammation. And I'm particularly sensitive to this one because this is an area on which we're working a lot. And I think it's really it is the 10th hallmark of aging. That being said, while that framework was helpful in organizing the field of aging research, we all recognize that as all approximation, as all hypothesis, it was only a framework. And so one of the things, for example, that I think was lacking is the understanding that aging is a multiform type of process that has many variables, and that, of course, you can get accelerated aging if you disrupt very significantly any one's of the hallmarks of aging. And we see this in human mutations. For example, if you have defective DNA repair, you're going to get accelerated aging. But that's an extreme that you see in some rare patients. And most of us, the understanding is that many of these hallmarks of aging actually are at work. And what is not clear is which one is the most important. How related are they to each other? Because one of the important point that you made at the beginning is the fact that identifying the targets also led us to the idea that if we target these processes, we're going to revert aging. And now we have drugs, for example, that will target, dysregulated nutrient sensing is one of the pathways that we work on. We have metformin, we have rapamycin that we know are calorie restriction mimetics. But the question that's not been answered is, how does this correlate to autophagy? How does this correlate to DNA repair and so on? We've entered a new phase in aging research where many people are starting to try to build links between these pathways, understanding how do they relate to each other. For example, when you use rapamycin, you not only affect nutrient sensing, but you also it's one of the biggest regulators of autophagy. So we published a paper recently showing that senescence, which is another hallmark of aging, is also associated with the dysregulated innate metabolism, which is linked to nutrient sensing. So I think we and many other people in the field are really busy trying to now understand the sort of former hierarchical tree of all these pathways and trying to understand how do they relate to each other. And most importantly, if you're talking about a network, which I think net aging is, where are the nodes? Where are the key molecules that we need to target to get the maximal beneficial effect? And I think this is where the field is going. So in this respect, the Rejuvenome, which is the collaboration that we have established with the Astera Institute, is aimed at this, is really tackling the second level of complexity of the aging field, really understanding the relationship between the different pathways and identifying the relative contribution of different interventions. How do they work in synergy, and where do you actually get the maximum synergistic effect?
It's so extremely important, and I'm as I said, when I saw this and I read about the combinations and the synergies, I was like, thank you, somebody's finally doing what needs to be done. So that was very exciting. And yeah, I mean, speaking of once again, going back to Hallmark for a second, we need to put, as you said, that chronic inflammation or inflammaging or whatever we want to term it in there. And I think I really take my hat off to anyone in the immunology space, and I think obviously we have heard so much the last couple of years about the immune system. Obviously, a focus of yours has been on how we appropriately balance this. We want a certain amount of inflammation. We don't want chronic activation of it, and then sort of outside of that axis, that we're learning, or you're learning, there's so much more. I mean, I saw a recent paper of yours, I think it was just a month ago, where you were looking at JAK inhibitors, which potentially can stimulate HIV reservoirs. We had a discussion recently on the regenerative medicine front about how certain components of the innate immune system are pro-regenerative. So we're constantly building on this unique inflammation immune system component of aging. Talk just a little. Obviously, the whole lecture's on this, I know, but could you sort of take us a step back and sort of how you look at this broader inflammaging domain as we sit here talking November 2021?
That's a great question, and I think it is one that I'm particularly excited about. And I think we are considering, giving its importance, actually building a whole new program at the BUC that's going to be focused on the biology of immune aging. And with a focus in humans. One reason why we are, what I think, I enjoy your question, and I think this is really highly relevant, is we are living it right now. The COVID-19 epidemic has highlighted in a way that no one could have anticipated what is immune aging. And COVID-19 has emerged as one of the major age-related immune diseases. Not so much being infected by COVID-19, but what is the outcome that happens to you? And we see this both in terms of, if you get infected, there's mortality, hospitalization, complications. Everything is correlated with age in an exponential manner. And also in terms of vaccination, which is even more interesting, because it turns out that vaccination rates decrease dramatically as you age. So even though that fraction of the population is at the highest risk, they also are the ones who are going to be the least likely responsive to the vaccine. So that really has brought back to the fore this idea that understanding immune aging is really important. So there's, if I can maybe just summarize what we know about immune aging, and why I think it is really important. So there are two arms of the immune system. One is the innate immune system, and the second one is the adaptive immune system. So the innate immune system, think about it as the first line of defense in our war against pathogens, essentially. And this is mostly macrophages, dendritic cells, and so on. These cells are present in all at the interface with the outside world, and are ready to start activating themselves and combating pathogens. And they do this in a very non-specific manner. They recognize sort of non-specific characteristics of pathogens. For example, double-stranded RNA is something that's present in viruses. So we have sensors that recognize double-stranded RNA. Now, this first line of defense is critical, because this is what allows you to build your army while the war starts. And what we know about this innate immune system, it's also not only recognizing pathogens, but it also recognizes damage. Mm-hmm. So if you have a cut, for example, your innate immune system gets activated, and this is what participates in tissue repair and wound healing. Now, associated with the aging process, we have increased damage across the whole body. And excuse me for 1 second, because my telephone keeps beeping. And so I should have done this beforehand. So as we age and tissue damage accumulates, and as senescent cells accumulate, there's a recognition that the innate immune system becomes chronically activated. And this is a process that people have referred to as inflammaging. Now, the consequence of this is that this chronic state of immune activation brings a whole series of secondary consequences. It is a key pathogenic factor in the development of a series of chronic diseases. So what is supposed to protect us eventually, when it's chronic and present constantly, becomes a problem by itself. And so the damage of aging induces the innate immune response, and then the innate immune response further actually is unable to fix the damage because it is linked to aging, and it eventually then becomes an increased risk factor for chronic diseases. So that's really the whole aspect of innate immune system. There's another problem which is linked to the adaptive immune system, which is much more selective. That's the idea. If you get infected by a bacteria or virus, your immune system will mount specific responses to that pathogen, both via B cells and T cells. And the B cells make antibodies, the T cells recognize the infected cells. And what we know is that that whole adaptive immune system becomes defective as you age. And it becomes defective for a whole series of reasons, and we can go on further. One of them is the thymic involution, so you lose the site of production of these cells and so on. So there's the adaptive immune response is also the one that's responsible for your response to vaccines. And so the long-term memory. So if you've been infected by chickenpox when you're a child, you actually keep a memory for most of your life for that virus. And that's all based on T cells and B cells. So the fact that this response is defective really has implication not only in your ability to mount a successful immune response against COVID-19, but also influenza. There are close to 40,000 people who die every year from influenza. And if you look at their age, there's a direct correlation with age again. So COVID-19 has brought this back. Something that's been under our nose for the whole time is that every single disease, that viral disease, is really it's the outcome is dramatically altered during aging. And I think no one has really addressed this or studied this, and we really want to start addressing, can we bring the science of aging to immunology, for example, to increase the efficacy of vaccination? Everyone, when you're in the vaccination field, everyone is interested in adjuvants and so on.
Sure.
I haven't heard anyone talking about the fact that we have novel drugs in the aging field that we could use to actually improve vaccination response. There was a brief attempt to do this by Restore Bio, you might recall. That used Rapalog to do this, and they had initial preliminary results that were very promising. Eventually, phase 3 trial failed, but I think this is a step in the right direction for a whole area of aging that is going to be critical in the future.
And continuing it, I think. I went into a lot of your papers, which are fascinating, didn't understand all of them, but one area that I would say maybe it's the 11th hallmark, or it's not on the SENS wheel of stuff either, is just this general theme of the virome, and that we are trillions of these little guys are within our system right now. I did an episode about a year ago with Jack Stapleton in Iowa, talking about this hepatitis G, which sounds really scary, but it protects us from herpes infections. And we're only learning sort of some of the beginning of the beneficial things that viruses can do for us, as well as how they harm us. anyway, I came across a couple papers. I mean, one is really fascinating me, and just this topic of gene drives, which whether we're talking about herpes, I know you published on cytomegalovirus as well. Talk a little bit just about sort of some of your visions to how, I mean, the gene drives are quite I don't know, call them a futuristic tool, but really cutting edge stuff. Some of your vision of how we could potentially use some of these new tools to maybe eradicate some of these viruses that we don't want, which are, sort of behind the scenes to this sort of chronic low-level inflammation that you were just talking about.
That's a great question. So I'll answer your question in two different ways. I'm amazed that you actually went that deep down into my CV to find a gene drive paper. We're quite excited.
It was a couple of years ago.
that whole project. So a word about herpes viruses first, which is really why we started working on this gene drive with a postdoc in the lab, Marius Walter. So herpes viruses are, there are, I think, 8 human herpes viruses. Mm-hmm. And it's been generally estimated that some of those are very viruses that we just talked about. Chicken pox, there's cytomegalovirus, Epstein-Barr virus, and so on. Yep. Many of these viruses actually occupy a large part of our immune system for most of our lives. Mm-hmm. And particularly cytomegalovirus, which has been shown, in many of us, actually, as you age, it can occupy pretty much 20 to 30% of all of your T cells. Is devoted to keeping this virus in check, because for all of these herpes viruses have the capability to becoming latent. That is, once you become infected, you actually do not cure the infection. You maintain the infection in a suppressed state using your immune system. And a good example here is also chickenpox, which is an infection of the children. It gets suppressed successfully, but it can reactivate when you are an older adult, and it can actually cause significant problems. Now, there are suspicion that many of the other herpes viruses, including herpes simplex 1 or 2 and 8 and 6, might be playing pathogenic roles in a number of chronic diseases, including Alzheimer's disease or multiple sclerosis or chronic fatigue syndrome. And I would predict that in the next 10 to 20 years, we will see emerging evidence that they are really, some of these viruses that actually affect most of the population are the drivers of some of the chronic diseases that currently we have no treatment or no understanding for. Mm-hmm. So I think it's a big area of virology that is occupied by a lot of people. I would not characterize myself as a herpes virologist, but we do work on a lot of these viruses because they're fascinating. Now, what is the idea of a gene drive, and why do we come up with this idea? A gene drive is a technology which is based on CRISPR and Cas9 technology, mm-hmm. which essentially allows you to disseminate a unique genetic cassette into a sexually reproductive population. And without getting into the detail, because it'd be really hard to do without a schema to show you exactly how does the gene drive work. Think about the gene drive as a dominant genetic element. Once mm-hmm. you introduce it into the population, it will spread, and it will spread very rapidly. And one of the classic example of a gene drive is one that has been contemplated. I don't know if it has been deployed, but to try to inactivate mosquito populations in areas, for example, where malaria is endemic or causing problems. You can actually introduce a genetic cassette into a founding mosquito, and then this mosquito will sexually reproduce and eventually will pass it to every one of its descendants. And eventually the cassette will eventually kill them. So that's the classical example of a gene drive, and it's been postulated by many people to be strictly dependent on sexual reproduction. So you need an animal that sexually reproduces, which essentially made it incompatible with the virus. But I had the good fortune of being approached by a postdoc from France, Marius Walter, who came up with the idea that there was really no reason why this should be strictly limited to sexual reproduction, and it should work for herpes viruses. And this is what Marius did. He built a gene drive that we can apply now to a herpes virus. And the way it works is you can have an infected cell where the virus would be normal virus would be replicating in an uncontrolled manner. If you introduce the gene drive in there, eventually it will slowly replace the wild type virus by the gene drive. Eventually, if you actually give some properties to the gene drive that allow you to control it, you can essentially replace the bad virus by a virus that is much more controllable. And so the first two papers that we've published with Marius in the last couple of years showed the fact that you can actually make this work in an in vitro system in a laboratory setting. And right now, I think Marius is quite busy doing this in the context now of an infected mouse. And I, without really, this is not published, so you can take this with a grain of salt, but the preliminary results look amazing. It actually does work in an animal model system. Now, the big question that's been raised is, in what clinical setting are we going to be, are we hoping to introduce these types of therapies? And I would say it's too early for this. We just need to learn how the system works. It's really highly experimental. It's not coming to humans in the next 10 years. I can guarantee you this. But it could represent a very significant new tool for a whole family of viruses for which we don't have good therapeutic interventions. So we're quite excited by that whole project.
Yeah, I wanted to bring it up just to sort of demonstrate. Obviously, you have an amazing CV, but just the scope of everything you're involved in is really fascinating. And I apologize if I'm making this more of a virology episode than aging. I'm going to come back to aging, I promise you, right after this next question. But I just got one other paper of yours, which was just really it lit the bulb. This is from back in February, but it was entitled "Evaluating a New Class of AKT-mTOR Activators for HIV latency reversing activity, ex vivo and in vivo." And obviously we talk a lot about mTOR on the show and rapamycin, but here we are, sort of, you're working in a holy grail area of latency reversing, which is sort of, the holy grail in HIV, and finishing up with that, hopefully. And here we have mTOR activation, not mTOR inhibition. Just say a few words about this paper, if you would.
Yeah, so it's actually a couple of papers that really, the first paper we published in the area showed that TOR is actually a critical regulator of the latency state of HIV. And the reason why why are we interested in HIV latency? This is another virus that can establish also a state of latency. Is that we know how to treat HIV today. We have combination of drugs. Again, this is a good example of a field where drug combinations proved critical in our ability to control HIV. And probably one of the first fields in medicine where the drug combination was instrumental in bringing down the epidemic. So we can now treat patients, suppress the virus. The problem is that the virus actually becomes undetectable, and we can treat people for a year or 2 years or 5 years or 10 years. The minute you remove the drugs, the virus comes back. Within 3 weeks, the patient is back at where it had started. And so this has led people to ask why, and it became apparent that there is a subset of cells in which the virus is dormant. So these cells are not sensitive to the antiviral drugs because the antiviral drugs only target the replication of the virus. And so this has led to a number of hypotheses or therapeutic trials of the idea that we should maybe try to force the virus to reactivate from these latent cells and then hit it with the antivirals. And so because of this, there was a lot of interest in trying to understand how is the virus controlled at the transcriptional level. It's all epigenetics. Again, we're going back to a field that is very overlapping with aging. And we actually, by the way, we're still working in that topic, on the topic of understanding the epigenetic regulation of HIV latency. It turns out we published a paper. We did a large screen using CRISPR, trying to find out what genes actually are controlling the state of latency. And the major regulator is TOR. But not in the way that we are used to in the aging field. It turns out that TOR activation is a critical pathway to reactivate latent HIV. And so this has brought the idea for people to try to use TOR activators or TOR inhibitors to modulate the state of HIV latency or reactivation. And so I live in both worlds, and but probably being in the aging field made me a little more sort of interested in when TOR popped up on our screen. I thought, well, this is quite interesting. Let's explore this.
Absolutely, yeah. That's what peaked. As I said, it hit the light bulb on that one, but that was fascinating as well. But okay, let's go back to aging now. Fascinating work in virology, nonetheless. But another major announcement. NIH a couple of weeks ago, through the Common Fund, they established the Cellular Senescence Network. $125 million, 16 grants, 5 years. Buck Institute, $12.7 million to Dr. Campisi and Dr. Schilling. And here we are again. Obviously, senescent cells extremely important, but we're beginning to realize that just like there's not one cancer, there's not one senescent cell, and we really need to understand these in a lot more detail. $12.7 million, a lot to get you there. Talk a little bit about the SenNet program, how you got involved with this, and a little bit of what they're going to be doing there in terms of further sort of characterizing senescent cells.
Yes. These grants are part of a process at the NIH called the Common Fund, which has a lot of significance beyond the award itself, because it is a special program that is carved out of the NIH budget. It is not run by individual institutes. It is run by the Office of the NIH Director. And they essentially pick areas that they think are highly promising to really accelerate research. And the fact that they picked senescence is a remarkable victory for the field of aging. As you know, we are considered a disruptive force in the whole field of biomedical research. It's a position that I relish, and I love to be the iconoclast and to bring novel ideas and novel therapies and novel solutions to medical problems. And the fact that NIH embraced senescence as a field that was worth investing $120 million, as you mentioned, is really significant for our field. Yep. So the vision of what they wanted to accomplish is really trying to further expand the realm and our understanding of senescence in humans. If I can say just a couple of words about senescence for your audience.
Please.
So senescence is a process that occurs throughout life. It's not restricted to aging, but it starts anytime a cell behaves in a way that is think about it as a genetic program that is activated by a number of stimuli. Some of those being, for example, if a cell starts activating an oncogene and it starts dividing in a way that's abnormal, senescence program can be activated, and it will put the cells in a resting state. It will force it to stop dividing, and it will also activate a process called the SASP, a senescence-associated secretory phenotype, which means it will trigger an inflammatory response so that the immune system can come and eliminate these cells. So we know that senescence, occurs also as a normal mechanism in wound healing, for example, in placenta, activation of labors. There's a number of processes in which we know that senescence plays a normal an important role. What happens during aging is that, at least this is the current thinking, is the removal of these senescent cells becomes impaired, probably because of immune dysfunction, as we mentioned. And this means that over time, the number of senescent cells slowly accumulates in most organs. Now, remember, I told you that these senescent cells activate a pro-inflammatory molecule that they secrete in the environment to trigger the immune response. So what it means is that as you age and as your burden of senescent cells increases, you are actually, this is thought to be one of the major contributors to the chronic inflammation. So they secrete inflammatory mediators like TNF-alpha, IL-6, IL-1, and so on. Now, out of this, and it's been documented actually, and the Buck Institute has Judy Campisi, who's been a pioneer really in this whole field, essentially, defining some of the key markers that really allow us to characterize these cells, to identify them. Also contributed to the founding of a company called Unity, and Unity was the first of the senolytics company. So once Judy and her colleagues, Jan van Dersen and others, had demonstrated that senescent cells actually play a key pathogenic role in the development of the chronic disease of aging, the idea came up that why don't we try to eliminate these cells with small molecules? And that David, who's a serial entrepreneur, actually approached Judy and came up with the idea, so why don't we actually target these cells for killing? And then the whole field of senolytic was born along with Unity, and they're still, going at it and trying to test these senolytics against a variety of age-related disease. So I think, does that answer your question?
Yeah. As I said, I wanted to, because we touched a bit on senolytics in the past, but I think I think what's really interesting, as you point out, $125 million, that's a nice investment for anyone, but for the NIH to be getting behind this and then really wanting to dive deep into sort of the details, let's say, of what is going on with this population of cells, whether it's in the ovaries, whether it's in muscle, whatever, is a major inflection point.
Yeah, I forgot my train of thought there, because that's true. You were asking me what is the program going to fund. So it's all focused on senescence, and their major goal is really to try to understand, to establish an atlas of senescence in humans. And so all of the funded groups are targeting different organs and really trying to understand where does senescence occur in humans, and in what tissues. And we picked a few tissues ourselves, and are going to be focusing on those as well.
Excellent. Eric, we've gone through a lot of your research in fascinating areas: immunoaging, senescence, the gene drives. You clearly have an amazing portfolio that you're working on. I just wanted to sort of turn to you've had a long experience as well, aside of doing all the research and active in medicine, working with industry. So you've been on the board of biotech companies. You an advisor to Sofinova Ventures and the venture capital scene, big French venture capital group. You've consulted for big pharma. What do you get as the current sense of where, what we'll call it, I don't know, what we'll call health span increasing, I don't want to say anti-aging, but where the whole basket is as far as pharma is looking at it nowadays? Obviously, a lot of VC money coming in, a lot of biotech companies. I sit in here in the sort of the pharmaceutical armpit of the world on the board of New Jersey. What are you getting as sort of the feedback from the Novartises and the GSKs and the J&Js of the world? Are they excited in 2022 about aging, about health span? What are you getting from that side of your network?
It's an interesting question, and it will. I suspect it will be different from company to company. Big pharma, I would say, is still looking at the whole field of aging research with sort of an intrigued but somewhat suspicious look. And I think it is in some way linked to a critical difficulty that we have in the field is, what are we trying to treat? And there's been a whole series of debate as to whether aging is a disease, whether it's not, whether it should have received its own indication for treatment. There's some people are pushing in this direction with the ICD from WHO. And there's a pushback, on the other hand, from a critical number of people in the medical field who refuse the idea that aging itself is a disease. I haven't made up my mind, by the way, in terms of whether it's really helpful or not to call it a disease. there's a part of me that feels that, if you're 80-year-old and you are really active and doing sports and in great shape, to be automatically called sick because you are 80 years old, it's somewhat disturbing. Mm-hmm. But on the other hand, there's there's the counter argument is that by categorizing aging itself as a disease, we might be able to do clinical trials against aging itself. Although I would argue also that the TAME trial that we know people are planning for with metformin is a true anti-aging trial, and it's not targeting a single indication. So there are many ways that we could go around the whole concept of aging as a disease or not. And in some way, for me, the debate is not truly helpful. The key question is really, what are we trying to treat? And there are a large number of identified modifications that happen during aging that have not been targeted in the past. For example, we were talking about vaccination. The lack of vaccination by itself should be an indication and could represent the basis of clinical trial. Sarcopenia, the loss of muscle mass that we see associated with aging, could be targeted. Thymic involution could be targeted. And the list goes on and on. There are frailty, which is this more terminal type of loss of muscle function and coordination. So this would be my preference, because if you want to target the pharma world and bring it into the field, their model is such that they will be more responsive to the idea of targeting really single indications, which, by the way, are much easier to conduct clinical trials on, macular degeneration and so on. So the hope is that when pharma starts conducting these trials, they will find out that some of the drugs that target aging pathways will actually have applications in multiple fields, and will target other diseases. It's a little bit what we've seen with metformin, where it was initially used, it still is used as a drug against type 2 diabetes, but it turns out, people who are on metformin are protected against Alzheimer's, against cancer, against heart disease, and so on. So metformin is really the classical example for me of how this field will sort of slowly infiltrate itself into the modern pharmacopoeia of that and big pharma. And I think, to me, this seems to be a more subtle way to make it into the mainstream than sort of a frontal assault by saying aging is a disease and we need to treat it as such. That being said, so pharma, I would say, for these reasons, is still watching this from the side. A good example was the fact that, for example, Novartis divested itself from Restore Bio when the idea of using rapalogs against age-related disease, they created a new sub-company, which to me sounded amazing, because if I had been Novartis, I would have tried to keep it in-house and build it as a big aging program. This didn't happen, and I think this is a reflection of what we are facing in the aging field. Everyone is really excited about moving all of our discoveries and our understanding of aging into the clinic. But being a physician and interacting a lot with friends and colleagues, I think we have a tough battle ahead of us. And confrontation, in my opinion, is not necessarily going to be the most effective way to get there. That being said, we're not only dependent on big pharma, and there's currently an enormous surge, and big pharma traditionally has been somewhat resistant to completely novel ideas, as all establishment generally is. And so one thing that we are seeing in the field is a surge of investment from VCs and angel investors and hedge funds. And so the good news, and donors, the good news is eventually we will get there. I can guarantee you big pharma will become interested, because they will realize the size of the market. And this is something that's affecting the whole population. The good news for us is that the money is starting to surge. And right now we have more ideas than we have resources. And I think the next few years, what we've seen in the last, just the last 2 or 3 years has been nothing but remarkable shift in terms of interest. And my prediction is that this will only accelerate in the future, and we're ready to ride that wave.
Excellent. Excellent. And you were just reminding me, because a couple of weeks ago, obviously I had Jennifer Garrison on from your organization, and she had also a very elegant, clinical development model there that once again, was focusing on a very narrow period of time in a woman's life where, we don't have to wait 20 years to see a result via X, but here is something that we can target, which I just wanted to lump that into sort of the Buck story, because I think it's another example of how, you're not just looking at things from sort of that longer term perspective, but there are some very creative clinical models that you're potentially getting involved in there, and that was another really exciting Buck story.
Yeah, menopause is another condition that we've always assumed that's just the normal fate of aging, but it turns out there are women who menopause very early, and we know this is a direct predictor of your lifespan. So that by itself becomes a medical indication. Maybe not if you are menopause at 60 years old, but if you're menopause at 40, for me, this would be a clear indication where there's a need to really understand what is happening and to intervene. So I think this is yet another example of what we talked about, sarcopenia, thymic involution, and so on.
Eric, before I let you go, we touched on virology, senescence, a lot about the immune system, clinical development, investment. Any other things coming up? Anything that I missed? Things early 2020, 2022 you want to mention? Please, I'll give you the floor to follow any follow-up messages, final messages for you.
Well, you catching me a little flat-footed, but one thing to realize is the excitement in the field, and the potential growth that is going to be happening. I think many of us have, there's such a thing as being too early in a field.
Sure.
And the Buck Institute has been in existence for now almost 22 years. And when it was founded, there was incredible excitement that, of course, the world was going to awaken to the amazing biology of aging. And that was happening, and it turns out that it took a long time. It's finally now, 20 years later, that we're seeing what many of us had predicted would happen: is that the biology of aging is entering a really exciting new phase of maturity, where big players are coming. We talked about pharma, but we see some of them actually indeed being interested. And looking at the field and trying to explore. So at the Buck, we've signed a number of large collaborations with big pharma, and so we see the interest surging. I think one word of caution is then once you move from animal models to humans, it is a tough battle. And I know there's an incredible amount of enthusiasm and excitement about what we can accomplish in animals, but once you move into humans, the barriers to success are much larger. And I've tried to stay on the side of caution by, some people have called me conservative or unimaginative, because I don't like to talk about a 200-year lifespan. I don't like to talk about immortality, about all of these aspects. We really are focusing on increasing human health span right now. And our goal is to increase human health span by 30 years in the next 20 years. And I think this is something that we're going to be able to do. That's my immediate focus. I would rather under-promise and over-deliver. And so if some in your audience find me somewhat conservative, is I know the world that we're trying to bring on board with us. And if we really want to change the world, I think we will have to bring some of the big pharma with us. There's no question about it. At the end of the day, they will be the one developing the drugs that will be massively distributed. That's the only way anyone has ever done it. There's no biotech that has done this by themselves, unless they've waited 20 years to do it. So right. I think I'm really optimistic about what the future will bring. but there's a lot of hard work to be done. And along with this, we also need a lot more basic biology. And right now, I mean, just to give you an idea, we have so many targets that need to be further explored. I would encourage your listeners, if some of them have the ability to invest or to support some of the research, there are more targets than we can go after. And this field will only realize its true potential when we can go after all of them and explore them independently and in combination, as you said.
Yeah. Well, you're clearly on the right path, and with everything we talked about today, I mean, the organization, the team that you have, obviously we've had some of the Buck folks on the show in the past, but you're really you're leading an awesome group there, Eric, and I just wishing you the best with all this moving it forward, and we will definitely help get the message out as much as possible through our own networks. For everybody that's going to be listening to this particular episode on the various podcasts or watching on the YouTube channel, you've been listening to the amazing Dr. Eric Verdin, President and Chief Executive Officer of the Buck Institute for Research on Aging, also Professor of Medicine, University of California, San Francisco. Eric, I want to thank you for taking the time out of your schedule to come talk to us. Thanks for everything you're doing there at the Buck. And as we like to say on our show, thanks for helping to create a better tomorrow for all of us via what you're doing. Really very impressive.
Thank you so much, Ira. I really enjoy your show as well. And I hope, this will have contributed a little bit to get everyone excited about what lies ahead.
Absolutely. Absolutely. Great seeing you.
Take care. Thank you.